Namaskar. In the last video lecture, we talked about mitosis. In this video lecture, we will talk about mitosis. Mitosis is a cell division that occurs in all somatic cells or body cells of an organism. It is a cell division that occurs in the multicellular eukaryotes. If we talk about plants, it occurs both in the sporophytic as well as gametophytic plant body. So as you know, these plants have sporophytic and gametophytic plant body. Sporophytic plant body is diploid. It is 2N. All the cells are 2N, whereas that of gametophytic plant body is haploid. So the sporophytic plant body starts with a zygote, which is 2N, and gametophytic plant body starts with a spore, which is haploid. So in both the cases, mitosis occurs. The number of cells increases by mitosis. In sporophyte, in fact, when the spores, before the spores are being produced, the meiosis takes place. So it will occur at a certain stage of the plant when the sporangium have developed the inner mass of tissue, the sporogenous tissue that forms the spore mother cell, that undergoes meiosis. Otherwise, all other parts of the body undergoes mitosis. In mitosis, the number of chromosomes or the sets of chromosomes remain same as that of the mother cell in the daughter cell. So if it is haploid undergoing mitosis, it will produce two cells having haploid number of chromosome. If it is a diploid cell undergoing mitosis, it will produce two cells which also have diploid number of chromosomes. Now let us see different phases of mitotic cell division. So mitosis or any other cell division will have two broad phases. Phase of nuclear division, that is called karyokinesis, and phase of cytoplasmic division, that is called cytokinesis. Karyokinesis comprises of four phases. Those are called prophase, metaphase, Anaphase, and Telophase. After Telophase, cytokinesis will begin. So each of these phases have the intermediate phases or transition phases. So they will have early prophase or pre-prophase, or early metaphase or pre-metaphase, like that, early anaphase or pre-anaphase, in this manner, because it is a continuous process. Cell division is a continuous process, although we define these stages, 4 stages in which the karyokinesis occurs. So before entering into the prophase, the cell was in the interphase. It was in the G2 phase. So if you see a cell cycle, in the cell cycle, say this is the these are the divisional phase. This is a divisional phase up to this. So the remaining is the interphase. This is interphase. Up to this. Interphase, as you know, comprises of 3 phases, G1, S, and G2. And the divisional phase comprises of prophase, metaphase, anaphase, telophase, and cytokinesis. So this is pro, meta, ana, telophase, and cytokinesis will take place. So if you see the morphology or the behavior of the chromosome during these phases, during different stages of the cell cycle, so if you begin from G1, if a particular chromosome in a cell, the chromosomes, all the chromosomes remain in a single chromatid stage. But when they pass through the S phase, during which the replication occurs, each of the chromosome now have two chromatids. Of course, they are very thin. The chromatids are very, very thin. They are not as thick as it has been drawn here during interphase. In fact, up to G2 or even in prophase, in the nucleus, the chromosome will look like very small, very fine fibrils. They will not be visible enough. So, like this, they will be entangled all along the nucleus. There will be numerous chromosomes. Here we are drawing in a simplified manner, just for our understanding. So now this will remain. The chromosome will remain in this condition up to metaphase, and after in the anaphase, the two chromatids, these two chromatids will get separated. One will move to one pole, the other will move to the other pole, and finally they will be in the telophase. There will be two different nucleus for the two chromatids. These chromatids are now chromosomes. So they again return into single chromatid stage. The chromosomes which were in a two chromatid stage, they are now again separated to form single chromatids. So they will again enter, each of them will enter into the cell division, and then they will have the same fate. So this is how the chromosome behaves in different phases of the cell cycle. So if you talk about the prophase, metaphase, anaphase, telophase, to begin with, the cell is here in the prophase, in the pre-prophase, when it is at the juncture of this G to M transition. What happens at that point of time? As we know that G to M transition is controlled by cyclin B CDK1 complex, and this complex, the activated complex, it controls, it phosphorylates three different proteins. One is called the nuclear lamina, the H1 protein, H1 histone, and the actin filament. Each of them have a different function. The nuclear lamina, it disrupts the nuclear membrane or nuclear envelope. H1 histone condenses chromosome. Chromosome condensation takes place. The chromosome becomes thicker gradually. Actin filaments are necessary to be activated because the cell undergoes morphological changes, and according to that, the cytoskeleton has to be reorganized. So actin filament will control that. So these three different phosphorylation events lead to the stages of that make the cell ready for entry into the prophase. So as we know, in prophase, the nuclear envelope disappears. So this is the nuclear envelope that is disappearing gradually. So regarding nuclear envelope in fact comprises of three things. One is nuclear pore complex, then the nuclear membrane itself, and the nuclear lamina. That is the layer of protein that is present just below the inner membrane of the nucleus. So each of them, the nuclear pore complex, it dismantles itself. The nuclear lamina, it depolymerizes, and about nuclear membrane, it is being said that either it is converted into very small vesicles which diffuses into the cytoplasm, or it is absorbed by the endoplasmic reticulum. The latter theory seems more feasible because the endoplasmic reticulum are in a continuum with the outer membrane of the nuclear envelope. So whatever the case, all these structures, they disassembled or dismantled in the prophase. So they disappear. The chromosome condensation occurs. So suppose this is the chromosome, so one of the chromosome I'm drawing here as a submetacentric chromosome, the other as a metacentric chromosome. So these chromosomes are getting thicker. Okay, and the nucleolus, which probably was present with say this chromosome, this is the satellite region, and here was this the secondary constriction that actually gives rise to the nucleolus. The nucleolus is basically, it's a place where ribosome biogenesis takes place. So it was there during the interphase, the whole interphase. But when prophase starts, the nuclear envelope disappears. It actually coils back into the secondary constriction. So it will coil back into this region. This is secondary constriction of the chromosome. Not all chromosomes have this structure, only some of the chromosomes in a cell will have secondary constriction, and that region is responsible for synthesis of nucleolus. That is called, that's why it is also called nucleolar organizer region, NOR. So these two things happen, the disappearance of the nuclear envelope and nuclear organizer region occurs during prophase, and the chromosome, they start to becoming thicker gradually because of the condensation, because of the phosphorylation of the H1 protein, in fact. And one more thing that happens is this, the centrosome that were present at one pole. Now there are two centrosomes, in fact, they were present, and they were in fact formed during the synthesis, during the S phase of the interphase. This centrosome has replicated during the S phase of the interphase. Now this replicated one of the centrosomes. There are two centrosomes in a here at this moment, but they will separate, and one of them will move to the other pole. So gradually this will become separated, and it will move to the, like this. This is microtubule organizing center. In case of plants, this area is not clearly visible, and there is a denser plate-like area over there that has been referred to as MTOC, microtubule organizing center, because it's centrosomes are basically, they are made up of microtubules, and they are the place of origin of spindle fibers or microtubules. So this moves to the opposite pole. During prophase, this movement starts, and when the cell enters into the metaphase, they have already reached to the opposite side. So here is one centrosome, here is another centrosome. Okay, so these centrosomes have started producing spindle fibers, also called astral rays. These astral rays, they determine whether the cell will divide symmetrically or asymmetrically. They actually they position the poles in the cell and the chromosomes as well. So when these spindle are growing, spindle fibers are basically microtubules. They are bundles of microtubules, and so the microtubules are made of tubulin protein, as you know. Tubulin, alpha and beta tubulin dimers, they assemble one after another in a ring-like fashion, and that grows gradually. So this end of the spindle is minus end, and this end is this end is plus end, and this end is minus end of the spindle normally, but it may change. So here the microtubules are getting shortened by the removal of the alpha and beta tubulin. Here it is growing further by the addition of these alpha and beta tubulin dimers. This is what happens in the poles. So this spindle fiber is gradually increasing in size, but it is in an oscillating position, that the size becomes a little bit longer, then it shrinks, then it again grows. So this goes on as if it is trying to find something in the cell. It is actually trying to find a chromosome, the kinetochore portion of the chromosome, that is centriole. The centriole contains there are trilamellar structure outside the centriole that is called kinetochore, with which the spindle fiber attaches. So the spindle fibers are trying to find some chromosome, the kinetochore of the chromosome. Say this is the chromosome that has bound to this particular spindle. So this will also associate with another spindle coming from the opposite side or opposite pole. Otherwise, the process is not completed. So in this manner, the chromosomes are distributed all through the nucleoplasmic region, and all of the chromosomes, they are bound by spindle fibers coming from both sides of the pole. This process is very crucial, and this is controlled by spindle attachment complex checkpoint that ensures that all the chromosomes are bound to the spindle, and this can be called pre-prophase. After this pre-prophase, the cell will undergo the prophase, in which what will happen, this chromosome has to be brought to the middle part, that is this part. This middle portion is the metaphase plate. It is the equatorial region of the cell. So all the chromosomes have to be brought to the metaphase plate before they get separated. Otherwise, equal distribution of the chromosome will not happen. So how these chromosomes are brought to the middle? It's that there is a tug of war going on over there. So suppose this is the chromosome that is attached with a spindle fiber. This is the spindle fiber that is being attached to it. On this side, this is the spindle fiber attached to it, coming from the other pole. So this pole is away from this pole, so it has to be brought somewhere here in the middle. So what will happen? There are proteins that will first of all, the length of this spindle will increase and this spindle will decrease. This will increase by addition of tubulin proteins on both the ends, but here the tubulin proteins will be taken off from this, so it will get shortened and shortened. And there are proteins that held these structures. There are proteins that are associated with the spindle fibers as well as the chromatids or the kinetochore. On this side there is a dimeric protein that is called kinesin 13. It's a motor protein of kinesin 13 family. On this side there is another dimeric protein of the same family, but it is kinesin, it is called kinesin 7. These are holding the spindle fiber and the chromosome together. This protein, the kinesin 7, is in fact, it is pushing the chromosome towards this towards equatorial zone, and this is pulling the chromosome towards equatorial zone, kinesin 13. On the other hand, there is yet another protein which is also a dimeric protein that is called dynein. It is working on the spindle fiber. It is attached with the with the chromatid, as well as it is bound to the spindle fibers. When the spindle fiber is gradually being shortened, the dimeric protein, it works along the remaining spindle, and gradually when this chromosome comes to the middle of the, say the chromosome has come to the middle of the cell now. So this is the middle of the cell. Okay, so at this position the tension on both the side will be equivalent, and this will be the signal that the chromosomes have come to the middle, and that will be the end of the metaphase. So in the metaphase, all the chromosomes are finally brought to the middle of the cell by this mechanism. So it will look like say as far as, and in metaphase also the centromeres have doubled, sorry, and the chromosome is most thickened during metaphase, so they will be like this. Okay, this is the metacentric one. The other one, which is submetacentric, will look like maybe something like this. Okay, and this is the equatorial plate or metaphasic plate. So this is the pole, and the spindle fibers are attached to the kinetochore like this on both sides now, and there are continuous spindle fibers also which have different role that I will tell you later. Okay, so this is the thing, this is called metaphase. Okay, now the cell will enter into the anaphase. So before entering into the anaphase, there is a checkpoint that is called anaphase promoting complex cyclosome checkpoint. What happens is these chromosomes during metaphase, they are basically, in fact, if you see them closely, the two chromatids, the sister chromatids, they are in fact these sister chromatids are tied together. They're wound by a protein that is called cohesin protein. These cohesin proteins, they cohese the chromatids, sister chromatids together. They bind the sister chromatids as if they will not allow them to be separated. So before anaphase has to begin, these cohesin proteins have to be removed, and that is being removed by the APC cyclosome complex checkpoint. This checkpoint induces a protein called CDC20, which is a ubiquitin ligase. So it ubiquitinates a protein called securin. Now securin is an inhibitor of separin. So when securin is removed, it is being degraded by ubiquitination. Then separin becomes, the inhibition on the separin is removed. Separin is actually a separase enzyme that will cleave these cohesin proteins. Okay, so this is securin, and it is bound to separin. This is separin. So when this will be removed, I mean, it will be degraded by ubiquitination and proteasomal degradation. Then this securin will be free to become active. So now it is active. When it was bound with the securin, it was inactive. So this active separin will cleave the cohesin, and now these chromatids are free of these cohesin molecules. The spindle fiber was attached to this, and it was pulling the chromosomes on this side. So now what will happen, this centromere will move apart, and at the same time the cohesin proteins have also been removed. So the chromosome is now not in tension. The tension generated on the spindle fiber is now absent. So that has been neutralized, in fact. So when this tension is almost null, the cell signals that the anaphase has begun and it can be separated. The chromatids, these chromatids will now be called chromosomes. When they become separated, these are no longer the sister chromatids, they are chromosomes now. So this is the anaphase. The anaphase can be divided again into two phases. Pre-anaphase, this was pre-anaphase, you can say, and then the post-anaphase. The post-anaphase or the anaphase itself can be divided into two phases, anaphase A and anaphase B. In the anaphase A, the poles, they remain at the same position. However, the chromosomes are moving towards their respective poles. In anaphase, in fact, you can see the shapes of the chromosomes that will tell you the position of the centromere, whether it is metacentric or submetacentric or telocentric or whatever. So in this way, these chromosomes are moving towards their respective poles. These two are moving on these two poles like this. This is the you can say this is anaphase A. Anaphase B, when anaphase B begins, then what happens? The continuous spindle fibers, those are there. The continuous spindle fibers will grow, will increase their length by addition of the tubulin proteins, and these two poles will be moved farther apart. They will move apart because of the increase in the length of the continuous fibers. And the chromosomes have come very close to the poles. Okay, so this is called anaphase B. So when this has been completed, the process of telophase will accomplish. So what will happen? The spindle fibers will gradually disappear, and these chromosomes have those which have brought to different regions, so two poles. In fact, they will be surrounded by a nuclear membrane now. So telophase is a stage when the nuclear membrane reappears. So this is the telophase. So after the telophase, it can be called telophase. Terminal phase of the karyokinesis. So as soon as the telophase is completed, the cytokinesis has to happen. Cytokinesis in animal cell has already begun during this anaphase. So that occurs because of the constriction in the plasma membrane in the central position, and that is being demarcated by a ring of proteins, the cytoplasmic ring of proteins that indicate the position where constriction will take place, and in fact they also assist in the constriction. So gradually this constriction increases. This constriction gradually increases. This is the constriction increases. There is a ring-like protein, that what I was saying, and gradually the two cell will become separate. So this is how cytokinesis occurs in animals. But in case of plants, since there is cell wall, the mother cell has a cell wall that has elongated because the cell was dividing. So there are two nuclei on both sides. Okay, then now the cell plate has to be formed. There has to be a cell wall in the middle. So it is formed. The process starts with the formation of accumulation of some Golgi vesicles as well endoplasmic reticulum vesicles over here. The accumulation of Golgi bodies and endoplasmic reticulum along with some proteins and microtubules is called Pragmaplast. Okay, and this place is also marked by the presence of microtubules. Okay, so now what will happen, these vesicles are like this. First these vesicles assemble there, and then they start producing projections which fuse with each other, forming a kind of a fenestrated structure. Okay, network like membranous structure, and that grows further by addition of vesicles. So here is the cell wall of the parental cell. Here is the cell wall on this side, here is the cell wall on the opposite side. So this will continue to grow on the lateral sides, and it will also deposit cell wall material, the pectic substances and the cellulose, hemicelluloses, etc., that will gradually deposited here in this fenestrated structure, this fenestrum, and gradually they will get thickened and solidified, and they will grow on both sides, finally touching the lateral walls. The membrane of the vesicles, these Golgi vesicles, they will form the plasma membrane. They will become associated with the mother cell plasma membrane like this. This cell had a plasma membrane inside, of course. So along with this cell plate formation, it has to be ensured that the plasma membrane is also formed inside these cells. Then this cell wall will fuse with the cell wall of the mother cell. Finally, this is the cell wall of the mother cell. So this is how cytokinesis occurs in plant cell. So finally, we can say that the mitosis has been completed, and mitosis involves these many steps. I try to cover everything on mitosis. If you find anything not being covered, or something goes wrong with your knowledge, then kindly put that in comment box, and suggest if you have some idea, some suggestion regarding this lecture. Thank you very much.